Hello everyone, thank you for giving the great opportunity of this presentation today. I am Akihisa Fukuda from Kyoto University, Japan. Today, I'd like to talk about the molecular mechanism of pancreatic cancer development. This is a picture with Dr. Mohammed Salah when he had a hands-on training fellowship program about EUS and ELCP at the Institute, Kyoto University Hospital, Japan, between June to December in 2018. Pancreatic ductal adenocarcinoma, PDAC, is one of the most lethal cancers. PDAC is the fourth leading cause of cancer deaths in Japan, and a 5-year survival rate of PDAC is only 7%. To develop novel therapeutic approaches, understanding the biology of the lethal disease is important. PDAC mainly arises from two premalignant lesions, Ipmn and Ipmn. Ipmn is a microscopic lesion. The majority of PDAC arises from Ipmn, and they have poor prognosis. In contrast, Ipmn is a macroscopic cystic lesions, and IPM-derived PDAC has relatively better prognosis for survival. IPM PDAC versus Ipmn PDAC is considered to be biologically different. However, the molecular mechanism of their development is unknown. Recent studies using mouse models have shown that Ipmn is derived from pancreatic acinar cells through the metaplastic state that is called acinar to ductal metaplasia. Here I introduce the Swiss-Smith chromatin remodeling complex. Swiss-Smith complex is a chromatin remodeling complex which changes the chromatin structure using an ADPase activity so that the transcription factors can access to their target genes and regulate genes expression. In this complex, we focused on BRG1 and RE1A. A recent study shows that Swiss-Smith complex is highly mutated in various cancer types, including PDAC. In fact, recent global mutational analysis shows that Swiss-Smith complex is one of the 10 mutation signatures of human PDAC. 14% of human PDAC have inactivating mutations in components of the Swiss-Smith complex. This slide shows significantly mutated genes in human PDAC. Among components in the Swiss-Smith complex, RE1A is the most frequently mutated gene in human PDAC. Moreover, interestingly, a previous study shows that loss of BRG1 expression is frequently observed in human Ipmns. However, the functional role of BRG1 in pancreatic tumorigenesis was still unknown. So, we sought to define the functional role of BRG1 and RE1A in pancreatic cancer development. First, we generated pancreatic-specific BRG1 knockout mice, crossing P2F3 KRAS mice with BRG1 flox mice. As shown here, pancreatic-specific loss of BRG1 with oncogenic KRAS results in formation of Ipmn, which progressed to form IPM-derived PDAC in mice. This is consistent with the findings that BRG1 expression is frequently lost in human Ipmns. IPM PDAC mice, P2F3 KRAS BRG1 flox mice, had much better prognosis for survival compared to Ipmn PDAC mice, P2F3 KRAS P53 flox mice. This is consistent with the human situation. RPM PDAC mice had much less metastasis and invasion compared to Ipmn PDAC mice as shown here. Because Ipm PDAC versus panning PDAC is biologically different, and panning is derived from acinar cells, we next asked what is the cellular origin of Ipmn. To investigate the cellular origin of Ipm, we crossed Keras Piaget flox mice with acinar-specific PT1-3ER mice or duct-specific H1B-3ER mice. We used the 3ER log speed system, which enabled to knock out BRG1 upon tamoxifen induction in a time-specific manner. As shown here, acinar-specific loss of BRG1 with oncogenic KRAS did not lead to Ipm formation. In contrast, pancreatic duct-specific loss of BRG1 with oncogenic KRAS resulted in Ipmn formation. These data show that Ipmn is derived from pancreatic duct cells but not acinar cells. So, BRG1 inhibits formation of duct cell-derived Ipmn and Ipm-derived PDAC. The next question is, what is the functional role of BRG1 in the formation of panning-derived PDAC? We next evaluated the formation of acinar cell-derived panning and PDAC using the acinar cell-specific BRG knockout mice model. We generated PTFI-3ER BRG flox mice in which BRG1 is deleted exclusively in acinar cells upon tamoxifen induction. In this model, recombination efficiency depends on the dose of tamoxifen. We assessed spontaneous ADM and panning formation in these mice. As shown here, significantly less ADM and panning were formed in BRG knockout mice compared to controls. This is the immunohistochemistry of BRG1. Arsenal positive panning shown in low panels is mostly BRG1 positive in BRG knockout mice. So, we concluded that BRG1 plays a critical role in oncogenic KRAS-induced spontaneous ADM and panning formation. Next, we examined whether BRG1 is critical for panning formation in the mutant p53 background. As shown here, panning was significantly decreased in BRG knockout mice compared to controls in the mutant p53 background as well. Again, BRG1 was panning is observed in BRG knockout mice or BRG positive escapers, indicating that BRG1 is also critical for panning formation in the mutant p53 background. Moreover, we assessed PDAC formation in these mice. All of 3 control mice developed PDAC in the BRG1 wild-type background. In contrast, only one of the 3 mice developed PDAC in the BRG knockout mice. Notably, PDAC developed in BRG knockout mice was only the BRG positive escapers. These data indicate that BRG1 is critical for acinar cell-derived PDAC formation in mice. These data demonstrate a critical role of BRG1 for panning-derived acinar cell-originated PDAC formation. The next question is, what is the underlying molecular mechanism? SOX9 has been shown to be a critical factor for ADM panning formation. Acinar cell-specific SOX9 knockout significantly suppress ADM and panning formation in mice. This is the same phenotype as BRG knockout mice. So we hypothesize that BRG1 positively regulates SOX9 expression and BRG1 SOX9 axis is the central mechanism in acinar cell-derived PDAC formation. To determine the functional relationship between BRG1 and SOX9, we investigated whether SOX9 overexpression cancels the phenotype of decrease in PEN information by BRG deletion. Remarkably, SOX9 overexpression cancels the reduction of PEN information in BRG knockout mice. From this data, BRG1 is critical for PEN information by regulating SOX9 expression directly. This is a short summary of the mechanism. BRG1 binds to the SOX9 promoter and recruits SOX9 upstream factors, including PX1, to the SOX9 promoter and induces SOX9 expression, which results in ADM and PEN information. In the absence of BRG1, the recruitment of upstream factors to the SOX9 promoters is compromised, and SOX9 expression is not upregulated, so acinar cell-derived ADN and PEN information is blocked. SOX9 overexpression accelerates ADN and PEN information and cancels the BRG knockout phenotype. BRG SOX9 axis is critical for acinar cell-derived PDAC formation. Notably, the relationship between BRG1 and SOX9 was also maintained in human panning and PDAC. Next, we analyzed messenger RNA expression from TCGA database. SOX9 messenger RNA expression level was also correlated with BRG expression in human PDAC. Next question is what is the role of BRG1 in maintenance and progression of pain? What happens when we knock out BRG1 in already established pain lesion? To answer this question, we used the novel flip-based dual recombination system. After panning was found, we knocked out BRG1 in pancreatic epithelium by recombination after tamoxifen induction. We aged animals up to 8 weeks to ensure PEN information and then deleted BRG1 by tamoxifen induction. Control mice had numbers of PENins. In contrast, BRG knockout mice had much less PENin, as shown here. These results show BRG1 is also critical for PENin maintenance. We observed increased apoptosis in BRG knockout PENins, as determined by cleaved caspase 3 staining. This is a summary schema. BRG1 plays a critical role in acinar cell-derived PDAC formation through regulating SOX9 expression. On the other hand, ARID1A is the most frequently mutated gene in switching complex in human PDACs. So next question is, what is the functional role of ARID1A in pancreatic cancer development? We next generated pancreatic-specific ARID1A knockout mice with oncogenic KRAS, PTM-free KRAS, ARID1A flox mice. As shown here, loss of ARID1A cooperates with oncogenic KRAS to form Ipmn. This is the same phenotype as BRG knockout mice. As shown here, cystic lesions in ARID1A knockout mice have characteristic of mucin connection to pancreatic duct and no ovarian stroma. Moreover, ARID1A negative Ipmns slowly progress to form invasive PDAC. Regarding to the cellular origin, similar to the BRG knockout mice, Ipmn was derived from pancreatic ductal cells, but not acinar cells in ARID1A knockout mice. Moreover, Consistent with the mouse data, Aridoine expression was frequently lost in human Ipmns, as shown here by immunostaining. This is a summary schema. Swiss-Smith complex chromatin regulator BRG1 and Aridone inhibit dark cell-derived Ipmn and PDAC formation. In contrast, BRG1 plays a critical role for acinar cell-derived penin and PDAC formation through regulating SOX9 expression. Our data highlight cell type specific roles of BRG1 in pancreatic cancer development. I thank all the collaborators. Thank you very much for your kind attention.